Okay, let's dive in. We have some really fascinating source material today on what looks like a genuine technological revolution in biology.
It really is, and it's aimed at a problem that's, well, it's fundamental. Biology is just staggeringly complex.
You mean systems like the brain?
Exactly. The brain, the immune system. You've got millions, even billions of components, proteins, cells, all interacting in real time in this dense 3D space. To understand any of it, you can't just look at the pieces one by one. You have to see the whole machine running.
And that's our mission for this deep dive. We're looking at a breakthrough that finally lets scientists map lots of proteins, we're talking high numbers, across a big, intact 3D piece of tissue, like a whole cubic millimeter of brain, all at once.
And for decades, that's been the impossible dream. Scientists were stuck with what we call the content-context trade-off. You had a choice to make.
High content or deep context, not both.
Precisely. You could get high content map dozens of proteins, but only on a super thin 2D slice. You see the players, but you lose the playing field.
Or you get the context, that thick 3D tissue, but you could only track maybe a few proteins. You lose track of most of the players.
So this research, it's all about smashing the two physical walls that created that trade-off.
Which are the color barrier and the depth barrier.
That's them.
Okay, let's start with that first one, the color barrier. The go-to method is fluorescence microscopy. We've all seen those incredible, colorful images of cells. So, what's the problem? Why not just throw more colors at it?
Well, it comes down to physics, really. When a fluorescent dye lights up, the color it gives off isn't a sharp point of light. It's a broad splash of light across a spectrum.
Okay.
Think of it like a radio dial. If each station's signal is really wide, you can only fit, say, 5 stations on the whole dial before they start bleeding into each other and you just hear noise.
So the colors literally just blend together, becomes a mess.
It becomes a mess. A common dye like CI5 has a huge spectral width. That's the color barrier. With the best gear, maybe you can push it to 7 targets, but that's pretty much the hard limit.
So to get around that, scientists came up with these clever workarounds like cyclic immunofluorescence or Cyc-IF. You stain a few things, take a picture, wash it all off, and then stain the next few.
Right, you do it in cycles. It solves the color problem, but it creates a whole new set of headaches, which is the depth barrier, especially when you try to do this with thick 3D tissue.
So what are these practical nightmares that come with cycling?
Well, the first one is just time, raw time. To get your antibodies to sink into a 1 millimeter thick piece of brain can take anywhere from 3 to 8 days.
Days for just one cycle.
For one cycle. So if you want to see 10 different sets of proteins, you're talking months. The experiment just grinds to a halt. It's prohibitively long.
Okay, that's a huge patience problem, but what about the tissue itself? What's the physical toll?
That's the second headache.
Damage.
You're repeatedly stripping, bleaching, and restaining this delicate piece of tissue. With every cycle, you're destroying some of the very proteins you're trying to see.
You're damaging the evidence while you're collecting it?
Exactly. Some studies showed that after just 3 cycles on a 1 millimeter sample, the fine structures were already starting to fall apart.
And I imagine even if the tissue survives, putting the data back together is tricky?
It's a registration disaster, a total nightmare. Every time you process the sample, it swells or shrinks a little bit. It warps in tiny, nonlinear ways. Now, imagine trying to perfectly align 10 different 3D images of this slightly different, wobbly object.
You can't. The errors would just stack up.
Which is why cyclic methods are basically stuck at the surface, maybe 100 microns deep. That is the depth barrier in action.
Which brings us to this new technique, radiant. The idea is to do it all in one shot. And the key is switching to something called Raman microscopy. How does that sidestep the color barrier?
The advantage is spectral narrowness. It's huge. Instead of seeing that big, wide splash of fluorescence, Raman measures the specific vibrations of molecules. And those vibrational signals are like laser pointers. They're incredibly sharp.
How much sharper are we talking?
About 50 times narrower. So that CI5 signal that was 700 units wide in fluorescence. In Raman, the same molecule signature is only 12 units wide.
Wow. So going back to the radio analogy, the signals are so tight you can pack dozens, maybe hundreds of stations onto the dial.
You've got it. It fundamentally breaks the color barrier.
Okay, but there's no free lunch. Traditionally, Raman is known for being incredibly weak. You get this amazing precision, but the signal is barely a whisper. How did they solve that sensitivity problem?
This is where it gets really, really cool.
Yeah.
They used an advanced method called APER-SRS microscopy. It stands for electronic pre-resonance stimulated Raman scattering.
That's a mouthful. What does it do?
I mean, in simple terms, it uses light to brilliantly amplify the signal from certain dyes. It's like a resonance effect.
And what kind of amplification are we talking about? A little boost?
We are talking about an enhancement factor of 10 to the power of 13.
Wait, 10? 10 trillion?
10 trillion times stronger. It's like turning a whisper into a Saturn V rocket launch.
That is not an incremental improvement.
Not at all. It's a leap. And it's the leap you need to get to nanomolar sensitivity, sensitive enough to see individual proteins inside a cell. So just like that, the main weakness of Raman, its sensitivity, is basically solved.
Okay, let's just pause here because that's a huge conceptual shift.
It is.
Recap 1. The big problem is the context trade-off, caused by the color barrier of fluorescence and the depth barrier of cyclic methods.
Right.
The new concept is radiant, using one-shot Raman for its super narrow signals. And the sensitivity problem is obliterated by EPR-SRS, giving it a 10 trillion-fold boost.
So we have sensitivity and we have spectral precision. But for you, the learner who's tracking this, have we solved the depth problem? Not yet. Because even with a loud signal, you still can't see through something that's opaque. Light scattering is still an issue.
Right. To see deep, you have to make the tissue transparent. You need tissue clearing.
Exactly. You couple EPR-SRS with tissue clearing using these high-concentration chemicals called refractive index matching solutions, or RIMS. But as you can probably guess, that created a whole new problem.
Of course it did. What was it this time?
They called it the Raman background pitfall. The clearing chemicals themselves, the RIMS, they generate their own really strong Raman signal. It's like turning on a giant floodlight right next to the thing you're trying to photograph.
It's chemical noise. Static.
A huge amount of static. And it just so happens to be the exact same part of the spectrum, the so-called fingerprint region, where most conventional Raman dyes operate. The signal from the dyes was just completely drowned out by the noise from the clearing solution.
So you solve the transparency problem, but you create a signal-to-noise disaster. So they couldn't use off-the-shelf dyes. They had to invent their own probes that were, what, invisible to the chemicals?
That's a perfect way to put it.
Yeah.
They engineered the Mars probes, Manhattan Raman scattering probes. And this is just beautiful chemistry. They're built around a special chemical reporter, a pi-conjugated triple bond.
Okay, why a triple bond? What's the magic there?
That triple bond vibrates at a super high frequency. It puts its signal way, way up in the spectrum, in a place called the cell-silent spectral region. And the crucial part is, in that quiet neighborhood, all the common biological molecules and, most importantly, all the RIMS clearing chemicals are totally silent. They don't make any noise up there.
Ah, so they didn't try to shout over the noise. They just moved to a completely different frequency where there was no noise to begin with.
That is spectral orthogonality. It's an incredibly elegant solution. The Mars dyes are essentially immune to the interference from the clearing agents.
So with that problem solved, they could build out their palette. How many targets did they end up hitting in a single shot?
They developed 8 distinct Mars probes. They tweaked them with different isotopes, like using heavy carbon, to shift each one's frequency just a little bit. Then they combined those 8 with 4 standard fluorescent dyes for a total of 12 targets.
12 targets, 1 image. That's the high content. Now for the high context, the clearing. They found some clearing methods were actually destroying their new probes.
Yeah, that was the next hurdle. The best clearing methods were solvent-based. But one popular solvent, using a protocol called rDISCO, was prone to forming peroxides.
And peroxides are nasty.
You're the enemy. They were just chewing up the Mars probes, destroying the signal. So they switched to a more stable protocol, rDISCO. But even that wasn't perfect, so they optimized it into their own version, rDISCO.
And what's the secret sauce in rDISCO?
2 things. First, they dropped the temperature down to 4 degrees Celsius just to slow down any chemical reactions. Right. But the real game changer was adding 1% propyl gallate, PG.
What's propyl gallate?
It's a ridiculously powerful antioxidant. I mean, about 1,000 times more potent at neutralizing those peroxide enemies than vitamin E, which is what people normally used.
1,000 times?
By adding this super antioxidant, they essentially created a protective shield for the Mars dyes during the entire clearing process.
And the results? The signal must have been much cleaner.
It was a huge jump. rDISCO gave them a 2.6-fold improvement in the signal-to-noise ratio over the next best method. And that performance held up even deep inside a 1 millimeter thick sample. The contrast was just beautiful all the way through.
Okay, so that's recap two. A lot of chemistry there.
A lot of very clever chemistry.
To solve the chemical static, they invented the Mars dyes to work in the cell-silent region, away from the noise.
Yeah.
And to protect those dyes from being destroyed, they developed the rDISCO protocol using cold temperatures and this super antioxidant propyl gallate.
Exactly. The full chemical toolkit was now in place.
So let's think about this for a second for the listener. When you imagine trying to analyze a millimeter of brain tissue, why is this one-shot approach of Radiant getting all 11 or 12 targets at once such a massive advantage over those old, slow, damaging, cyclic methods?
It's all about spatial fidelity. Because you capture everything at the exact same moment in time in the exact same physical space, the 3D map is intrinsically perfect. There's no warping, no shrinking, no registration errors. What you see is what was really there.
That structural truth is the foundation for everything else. Which leads us to the big payoff. What was the final record? What did they achieve with the full Radiant and rDISCO system?
The breakthrough moment was getting a one-shot, 11-target protein image across an entire 1 millimeter thick slice of a developing mouse cerebellum.
A full millimeter.
A full millimeter. That's 10 times deeper than the best cyclic methods and over 100 times more volumetric context than mass spectrometry. It's content and context finally together.
So they have this incredible 3D data set. What did it actually tell them about the biology of that developing brain?
Well, first they did the basic sanity checks. They segmented the image into the cerebellum's 4 known anatomical layers, and they ran correlation maps. And you see what you expect. Nuclear markers are correlated in the dense cell layers.
And things that should be separate are separate.
Exactly. The astrocyte marker and the neuron marker show negative correlation. So check 1. The technology is faithfully mapping distinct cell types in the right places.
But the real power comes from looking at processes, like development. They looked at astrocyte differentiation, right, using 2 markers, vimentin and GFA.
Yes, and this is where the 3D snapshot becomes so powerful. They could identify 3 different cell populations. The progenitors, the ones in the middle of differentiating, and the mature ones. And what they found at this specific age, P15, was that the vast majority of astrocytes were caught in that middle stage, co-expressing both markers.
So they caught them right in the act of developing.
They captured a biological moment, a peak in early glial development that happens to coincide with a burst of myelination in the brain.
And because it's a perfect 3D map, they could ask a really critical spatial question. How close are these developing cells to the blood vessels?
Which is vital for understanding the blood-brain barrier. So they measured the 3D distance from every astrocyte to the nearest blood vessel. The average was about 7.8 microns.
But I'm guessing there was a more interesting detail in there.
There was. The cells that were closest to the blood vessels, just 5.9 microns away on average, were those differentiating astrocytes, the ones caught in that middle stage.
What does that suggest?
It suggests that there's this peak of coordination between the developing glia and the vasculature right at this moment, which is likely critical for building a healthy, functional blood-brain barrier. It's a spatial relationship you could never see in 2D.
Incredible. So let's zoom out one last time to the entire network. What did they see when they connected all 11 dots at once?
They saw what's called a small world network. Basically, everything is highly connected and efficient. Any 2 proteins in the network are only separated by 2 or 3 steps.
Highly efficient communication. And were there key players, hubs in the network?
There were. Overall, things like blood vessels and astrocytes were central hubs, which makes sense. But the context mattered. A protein called TUB3 was only a major hub specifically inside the Purkinje layer.
And that means something biologically.
It's a perfect confirmation. We know TUB3 is essential for maintaining axons in Purkinje cells. Radiant didn't just find that connection. It mapped its importance to a specific 3D architectural location within the system.
So the final takeaway here is that this isn't just a small step forward.
No, Radiant has genuinely circumvented the decades-old content-context trade-off. It's the combination of the one-shot enhanced Raman with the custom-built chemistry, the Mars probes, and the rDISCO protection.
Now, for the listener, we should be clear. The researchers point out it's not perfect yet. That big 11-target image took about 4 hours, so the next step is speed.
Right. They're looking at ways to amplify the signal even more to speed up the imaging and also to expand the palette beyond 20 targets.
And the ultimate goal, the grand vision,
is to make Radiant a spatially resolved panomics tool. To not just image proteins, but to simultaneously map RNA and DNA in 3D as well. To see the entire system's state genome, transcriptome, proteome, all within the intact tissue.
So for you, the learner, now that you're up to speed on the absolute state of the art here, we just heard that the differentiating astrocytes cluster tightly around blood vessels, likely to coordinate the building of the blood-brain barrier. Thinking about that, about how critical these precise 3D spatial relationships are for healthy development, what other complex neurodevelopmental processes, maybe ones that go wrong in disease, could a tool like this finally allow us to see and understand?
We're finally truly moving beyond flat maps. We're starting to explore the real, complex, three-dimensional architecture of life itself.